everyone, welcome to our channel MedVeda, where you get a new learning experiences with me. And today our topic is erythropoiesis, which is the continuation of our previous topic, erythropoiesis, in the previous video. The link of the video is given in the description below. Please click on it and watch it. Now we will go into our topic, regulation of erythropoiesis. Regulation of erythropoiesis is influenced by various factors. We have categorized them into two main factors, general factors and special factors. These special factors are further divided into gastric intrinsic factor, extrinsic factor and dietary factor, and the general factors are divided into hypoxia and hormones. And now we will go into one by one topic in a detailed way. General factors, this includes the overall view, and we will discuss about hypoxia. As we know that hypoxia is due to reduction in the oxygen or lack of oxygen to the tissue or decrease in the oxygen supply to the tissue. There are factors that causes the decrease in this oxygen supply, like low blood volume, anemia, low hemoglobin, poor blood flow, pulmonary diseases. This causes the reduction of the tissue oxygen, and this reduction in tissue oxygen causes releases of erythropoietin, which is important hormone to influence the erythropoietic process. And most of this erythropoietin is released by the kidney, about 85%. Remaining 15% is produced by the liver and macrophage tissue cells. And this erythropoietin is mainly produced in the kidney by the JG cells, and this erythropoietin released goes to the hemopoietic stem cells in the bone marrow to release proerythroblast cells. Not only act on the hemopoietic stem cells to produce proerythroblast, but it also acts on all the stages to increase the production of RBC by releasing the mature RBC into the peripheral circulation to compensate the depleting oxygen. Next, we will discuss about hormones. Hormones also plays an important role in the regulation of erythropoiesis, and some hormones may increase the production of erythropoiesis, some may decreases the production of erythropoiesis or decreases the rate of erythropoiesis. Hormones like thyroxine, androgen, anterior pituitary hormones like thyroid stimulating hormone, growth hormone, adrenocorticotropic hormone, luteinizing hormone, follicle stimulating hormone, prolactin hormone, these all increases the RBC production, whereas hormones like estrogen decreases the RBC production. And this is one of the main reasons why females have less RBC than compared to the male. Now we will discuss about special factors. Special factors consist of gastric intrinsic factor, extrinsic factor and dietary factors. These dietary factors are including iron, protein, nickel, copper, cobalt, vitamins like riboflavin and nicotic acid. This helps in the production of hemoglobin. Whereas the gastric intrinsic factor, it is produced in the stomach in the gastric juice. It helps in the absorption of vitamin B12 in the small intestine. Without gastric intrinsic factor, vitamin B12 cannot be absorbed into our blood, so this causes megaloblastic anemia. Extrinsic factors, vitamin B12 and folic acid taken in the diet helps in the development of RBC by condensing the DNA and maturing the DNA. Without it, both maturation and condensation of the DNA doesn't occur. So it is all, these two also plays an important role in the regulation of erythropoiesis. We will know the fate of the red blood cell in the circulation after it completing its lifespan of 120 days. And first we have to know where the destruction occurs. Where is the place where the destruction of the RBC takes place? Mainly it occurs in the red pulp of the spleen and liver, and sometimes in the bone marrow. Now we have to know, we know where the destruction occurs. Now we have to know what is the process, by which process the destruction takes place. As we know that after RBC, after 120 days, it becomes very fragile, that when it's passing through the capillaries, its membrane may rupture and release its contents into the blood, which attracts the phagocytes, which phagocyte is this RBC, and bring them into the spleen, where they break down it, and the hemoglobin is break down in the macrophages, which forms heme and globin. We know that globin is a protein. It is further breakdown into amino acids, which can be reused to produce the hemoglobin in the bone marrow. And the heme is further breakdown into iron and proporphyrin. And the iron, which is produced as it easily produces free radical, it is bound to the apoprotein in the spleen, and from there it is released into the blood. In blood, the free iron binds to the transferrin. This transferrin brings the iron to liver, where in liver hepatocyte cells which takes this iron, and in hepatocytes this iron binds to apoferritin, and this converts the iron to ferritin, which is a stored form of iron, and this stored form of iron can be reused in the production of hemoglobin. Next, porphyrin. What happens to the porphyrin? This porphyrin is further breakdown into biliverdin, and this biliverdin is further breakdown into bilirubin. This bilirubin is released into the bloodstream, which is unconjugated, and it is not very good for our body. And this bilirubin is bound to the albumin to prevent its harm, and from albumin in the blood it carries to the liver, where hepatocytes takes this bilirubin and conjugates this bilirubin in the hepatocyte cells. Their conjugation bilirubin occurs in the hepatocytes. This conjugated bilirubin is released into the small intestine along with the bile juice, where it is excreted in feces in the form of stercobilin, and where there is 5% of absorption of this bilirubin from the small intestine, which enters into the plasma, from there into the blood. When blood is filtering through the kidney filters this bilirubin, and it is excreted in the form of urine in urobilin form. Thank you for watching this video. Please stay tuned to our channel for the further videos, and please like, share and subscribe our channel. Thank you.